All right, so I greet you wherever you are. You're welcome to today's episode. Today, we want to look at principal component analysis. And let me share my. So, we are going to be looking at principal component analysis. What is it in R? But my R studio is a little bit freezing, so please, I don't know what the problem is, but it's a bit freezing. So what we have here, so as you can see, it's not properly working like the way it's supposed to be. It's a little bit of freezing. So we are going to do principal component analysis, and it's all about dimension reduction. One of the easiest way to be able to make sure you normalize data when you are doing data analysis is principal component analysis. So what it does is it actually reduces the dimensions of the data into a much more data variable, which are quite related, or the variation in them is not that much. So that's what principal component analysis does. So in the mass package, there is a data set called biopsy. Let's run that and see what is inside. And so maybe I need to load in the mass package. And so I'm going to run that. So I've loaded my mass package. So let's see what the biopsy is all about. So I'm trying to print out the biopsy data. And then, yes, when you put a question mark and you run against any data set, it's an inbuilt data set, you're going to have the description of the data as you can see it around here. So I have the biopsy data. Biopsy is actually a sample. It's like one of the ways of collecting a sample. And then we see that we're going to have something like ID. These are the columns or the variable names. So we have the ID that's the sample code number. And then we can have the drunk, what the clamp thickness. We can look at the uniformity of the cell. That's for column 2. Column 3 will be uniformity of the cell shape. And then column 4 is marginal. And then we can look at all the columns. So let's just look at the first 6 rows of the columns that they are referring to. So this is actually a cancer data set. So you have the ID representing the individual person that the sample was taken from. And you can see all the description of those particular cancer cells. You have V1, V2. So if I have V1 here, you should know what V1 is about, the clamp thickness. Then the V2 is about uniformity of the cell size. And then you also have uniformity of the cell shape. That's our V3. And then marginal adhesion, that's our V4. And then you look at the description. And then the last column, which is the class. So it tells us the class of the data of the cancer we are talking about. Either it is a benign cancer or malignant. Benign means the cancer is stationary. It's not moving. Malignant means it is a spreading cancer. Some cancer, when it affects one part of the body, for like it spread to the other part of the body, and that's malignant. So we want to use this data to do a principal component analysis and see how it looks like. So let me, like I'm saying today, my R Studio is freezing. I don't know what the problem is, but it's crazily freezing. And so let's look at, first of all, if you look at you have to look at the structure of the data, which is very important. And then the structure, it tells us that it's actually a data frame. As you can see, it's a data frame here. And then we have 699 observations of 11 variables. So we need to know that. And then let me just slide this down. The next thing is you check the dimensions of this. Also going to give us a data set of 699 rows and then 11 columns. And of course, I'm going to jump this line. So the first thing you have to do in principal component analysis, you need to ask whether in that particular data set, do you have NAs, that like data that was not picked? So I'm asking the question, do we have some NAs in that particular data set? And then it's going to return a Boolean. And that's where we are seeing false, 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 false. It means if there were NAs, if there were some missing data, then you would have seen true. But as you see false, false, it means we do not have any missing data. But to be on the safer side, just make sure that you create a new variable and omit all the missing variables. So we are saying that ignore all the missing variables. This is not the best way to do that. We use a time reverse to be able to replace the the unavailable data with maybe the mean of the data. I'll do that in how to do data wrangling. Watch out one of my video tutorials on that. And so now we have omitted if there is any NA, even though we've seen that there's no NA, but you see that we couldn't print all. We printed up to 600 and we printed without 609 rows. So that's why I'm just assuming that there could be some NAs hiding somewhere, but maybe for the first rows that we have printed, we do not have any NAs there. So that's why I returned this. And so I just decided to omit it. So we come here and we have to also make sure that we omit all the categorical data. So the categorical data here means when we looked at the structure of the data, there was something that we needed to check. When we looked at the structure, you notice that. So you see that the class here, they are factors, so they are categorical. If you also check the first one, the IDs, they are also strings, so they are much more categorical. So the rest, we see them, they are integers, and then they are all integers. So we need to omit column 1, and then we also have to also take out column, the last column, 11. And so that's where we came here, and then we ran this particular code. And what we are saying is we go into the biopsy, and then we are picking all the rows, but we are exempting column 1. That's why we say -1, and then -11, and the last column, which is -11. So we ran that, and we pack the rest of the data into what we call biopsy, ending with this. So I'm just going to run that, and so I have my biopsy now. So when I do, what is it? I'm going to convert it from the data frame into a matrix. So, and then I'll create it as a new variable called here matrix, and so we need to check. Why I am doing this is that we need to check the eligibility of the data, whether the data is eligible for principal component analysis, because it's not every data that you can use it for principal component analysis. I need to be clear about that. And so to be able to check the eligibility, you need to check the mean correlation. You find out the average correlation. If the average correlation is above 0.3, and then it's also not below -0.3, then it means the data is good. So if you pick any of them, so it's just within that particular range. From 0.3 and above, the data is good. So we have to print out the correlation and then go ahead and print the correlation average or the mean correlation. So to be able to print the correlation of the data, we need to convert it from the data frame into a data matrix. And that's what we are doing here. So we converted it into data matrix, and then we look for the correlation. And so we decided to do the correlation by using the call function of the matrix we have already and pack the variable into biopsy correlation. And so we have that. So when we print this, we can see all the correlation figures which are here. But that's not what we are looking for. We are looking for the mean correlation. So once we have the correlation inside here, then we use the mean function to find out. And so we notice that the mean function says that it is 0.6. So once it is more than 0.3, it means the data is eligible for principal component analysis. And that's all that I was trying to put over there. Now, the next thing is for us to perform the principal component analysis. And we're going to use the function called the pre-comp function. And then that's what we're going to use. So we print our first, and then we are standardizing all the data. That's why we are setting the scale to true. So it means we don't want any bias. So we're setting everything to scale. And so this is what we are running. And so our principal component analysis is here. That's why I say biopsy _ PCA. And that's all about it. So let's look at the downstream part of it. So the downstream part, we're going to have analysis of the component. And then, so first of all, let's look at the summary. The summary statistics of the principal component that we have printed is over there. And so let's run the summary. And then we can look at how many components do we have. There are about 9 components. And we can look at the standard deviation, the proportion of the variance, and then the cumulative proportion. And so the standard deviations are very important. If you want to look at, after you have printed the principal component, sometimes you want to look at the variables that the most important variables which are there. So you use the name function, and then you run it, and then to print. So we have the standard deviation, we have the rotation. The rotation is the same thing we call the eigenvalues. And then we have the center, which means the mean. We have the scale, which is also the standard deviation. And then we have the X, and I'll be talking about each of them. So to print out any of them, if you want to print out the standard deviation alone of of the principal component analysis of each of the component. We can see that in component 1, this is the standard deviation, and then we know what standard deviation is all about. And then we can print the eigenvalues, which is very important. Yeah, so this is the eigenvalues for all the components. And if you want to know what eigenvalues are, check my next video. I'll be explaining what eigenvalues are all about. We can check that for the center, and then of course we can also check that for the scale. And you notice that the scale, as you can see around here, 2.820761. Then let's go here and check. All right, so now let's look at the X. The X, so these are the components of the principal components that we just printed. And so that's the principal components itself. So it's going to print the principal components of values for all of them, so which we have already seen it before. And so this is what we see. That's the principal components of all the component that we are looking at. And so that's what it's going to summarize. It has reduced the. And so now let's check something. Let's look at how to be able to visualize the principal component analysis. So how do we do that? We use a function called the fvish_ig. So it's a function that is found in a package called Factor Extra. If you don't have Factor Extra, maybe you may want to install it. I have Factor Extra, so what I'll simply do, I'll just load it, and then I'm going to run this particular library to load my Factor Extra. So that way, it gives me the opportunity to be able to use this function to visualize my principal components and how it is going to look like. It's very okay. My array is freezing. I've already mentioned this. As you can see, I'm trying to shift this a little bit, but it's not just shifting. Yeah, it's freezing. It's so bad. So you need to just pardon me for that. So I'm going to run this line 206, but you see that I'm unable to move because it has freeze. So when we run that, we are trying to look at the first visual of the component, and we'll look at how to be able to pick out the component that we can use for our downstream analysis. I've run that, but as you can see, it's taking a long time before it's going to print because my array is freezing. And when you see this red thing here, it means that there's something going in the background, just that the result is not yet out. So I think it has run successfully, but we just can't see the plot because it's taking time. Yeah, the plot is out now. I'm trying to zoom it for us to see what it means, but as I said, it's freezing so badly. So this is the first part. So we can look at how the dimensions are. And so there were about 9 components. And so you can see component 9, which is here. And you can see the similarity percentage, about 1%. So if we check, we have about 69.5%. So if we are going to go for further analysis, this is the component that we would have considered component 1, which is normal of all principal component analysis. If you are going to add another component for downstream analysis, then you would have considered the second one. Obviously, wherever this particular line that you are seeing here, the script plot, this is a script plot. So wherever you see this bend, where the elbow is, that is where you should stop. So it is component 1 and 2 that we should involve in our downstream analysis. Obviously, from 6% similarity, if the dimension reduction has given us some 6% of the data variable which are here, I don't think it's quite that necessary. So we consider component 1 and 2 in our downstream analysis. So we will go ahead and then, that's for the first. We could actually do a biplot. We do a biplot with default settings, and we run that. And then that is going to print the biplot for us. And that way, it's going to divide the data into, or it's going to it's going to give us this kind of data with the data variables which are also over there. Because we run it with default, the data variables will be there. And as you can see, And so we can see the clustering, which is here, or sorry, we can see the component, which is here, all the variables here, what they seem to have the dimensions. There are those who have practically the same what attributes. And then you can see other components here, which are also scattered all the way up to this place. But this is a default. Then let's actually add some labels. And then we are going to say that label, we run that. Let's see how that's going to work. I would have finished this tutorial by now, but it's so bad. It's freezing, so badly freezing. I don't know what the problem is. And so here we have given our own labels, and we are saying that we want the labels to be based on the variables. We've already seen what are the variables. So we will see whether it is a clam something, whether it is the size, it is the shape. So we can see it here. If we zoom it out, that out, it's taking a whole lot of time. Yeah, so we can see what is it? So you can see variable 9, V9 is around here, and see V5, and then in that order. So we label the data based on the variables, and then that's also another way of doing that. And here we are going to do the biplotting based on groupings. So we are now going to group all these data points based on whether it is a benign cancer or it is a malignant cancer. And then, so this is how we're going to write the code. And we are saying that the label will be based on the variables, and then the habilage will be based on the class. Remember, the class gives us whether it is a benign or it is a malignant. So it will group the data based on this. And then that's why we are using this argument called habilage. So we run that and see what is going to happen. So let me close this a little bit, and then you see. It's freezing so badly. It's freezing. So it has run, but you see that it's taking quite a number of minutes. And as I'm sitting, I cannot move anything. Yeah, you see how it runs. So it tells me an error somewhere. I cannot pop a two-level viewpoint, grid point. So let me check where the error is coming from. So it's saying that it's loading error in grid.call.graphics. Let me see. And so I have based on groupings, and then this was what I ran. So it's taking unnecessary time. But if it doesn't run, I think I need to activate. I need to load in my ggplot2, and then I see how it's going to look like. Yeah. So now we can see that our principal component. Yeah, our principal component is here. It's now based on it's based on what is the benign. So wherever you see the round shape, it means that's a benign cancer. And then we can also see malignant. So we can see all the benign grouped here, and we can also see all the malignant, much more grouped here. But you can see the malignant are also spread to other kinds of what are the other dimensions, as you can see. And then that is it. So let me run the very last plot here. And so here we are plotting based on grouping using customized colors. Here, I just want to choose the colors that I want. And then let me explain the code before. So here we are saying that we are using this particular function called the fv underscore PCA underscore biplot. And we are using this, the PCA we have already built, which is biopsy underscore PCA. The label we are using the variables, the habilage, that's we have already explained the habilage on top here. That's we are using a class based on whether that's the groupings, whether it is benign or it is malignant. And we are saying the color dot var is going to be red. So we are talking about these lines here. The lines you are seeing here, the arrows here, they are going to be red colors. And then we use the plus. When you see the plus, it means we're using the ggplot function or a package to be able to plot this. So we're using a particular function, the ggplot, called scale_color_manual. And then we simply use values and then create the colors. Once the groupings are 2, we have to create 2 colors. And that's what we see here. So when we run that, we're going to have green for benign and then blue for malignant. And that's just all that we have done there. It's taken the same way. It's taken a bit of time to be able to print that out. And so if we didn't also, so there you are. So you can now see that we have our groupings, which is based on benign, which is green, and malignant, which is blue. And so we can see that here we can see this is the benign cancers. And then we can also see that the malignant are also here. And so we can see the shapes are even not the same. But somehow we also see some infusion of the benign cancers, which are also here. And we also see a few infusion of the malignant cancers, which also groups here. Maybe it means they have some level of similarity or have some kind of similarity around here. And then that is it for principal component analysis and how to be able to visualize it, and it makes a little bit sense from it. Sorry, my computer is freezing today. I don't know what the problem is, and that is why the tutorial has taken this long. I'll try my possible best and fix that. Before the next video, I'll be looking at binning, because the idea is to get to this particular place. Let me pull this out and then I show you what. Here, we're driving to this place. That's machine learning. We're going to have supervised and reinforced learning. And of course, under the supervised learning, we have algorithms, and then we have so I've already done decision trees. Random forest and decision trees are not too different. And then that's what we're looking at, classifications. And then I think I've done some bit of classification on that videos on my YouTube channel. And if you also come to look at unsupervised learning, one of the algorithms that is used is either PCA, and that's why we are doing the PCA today, as you can see it here. It comes under clustering. The algorithm is clustering, and the type, it can be PCA, K-means, and then SVD. And so that's why we use 3 here, and here we are using PCA. And so we will do this before we come to machine learning itself. So thanks for being around. I'll try my possible best, and then I continue to upload. Share it for other people to also enjoy. Share it on your platform. Make a recommendation that you think is necessary, and then I will simply go by it. Thank you. My contact is here.